Okay, so welcome to this next video in which we are discussing non-homologous end joining for the repairing of double-strand DNA breaks. Okay, so in the previous videos what we've discussed is the detection of double-strand breaks, and we've discussed that this leads to the activation of p53 transcription factors, and once active, p53 transcription factors are going to increase the expression of proteins involved in DNA repair. Okay, so most of the proteins that we're now going to discuss in non-homologous end joining are going to have their expression increased by these p53 transcription factors. Okay, so let's now discuss non-homologous end joining then. So firstly, let's redraw out our DNA which has been broken. Okay, so here is the double-strand break in our DNA. And remember, we've discussed that the double-strand break in the DNA doesn't need to be clean. It can be fragmented with these overhangs, which can be on the same side, most unhelpfully here. Okay, right. Now, what's going to happen is we have seen how mRN complexes will come and associate with these free ends that we have here, both of them, and then you'll get ataxia, telangiectasia mutated serine threonine kinases associating with the mRN complexes. However, those will occur very, very quickly, but you're also going to have other proteins associating onto these free ends that are going to actually be involved in the non-homologous end joining. These tend to take a lot longer to actually be put on, mainly because most of their proteins that are going to be involved in these complexes are going to actually be expressed once the p53 has actually become activated. Okay, now I am not going to show the mRN complexes still bound here because it will just make the picture look even more complicated than it's going to be anyway. So we're now going to have a clean picture here, but you should remember that the mRN complexes have not suddenly vanished, they are still there. Okay, so let's now start looking at the first protein that is going to associate in the non-homologous end joining machinery. Okay, and this is known as a Ku protein heterodimer. Okay, and this is going to be a dimer of two proteins that are called Ku proteins. Okay, so it's going to have two Ku proteins in. One is called Ku70 and the other is called Ku80. Okay, and together, when you assemble Ku70 and Ku80 together, they're going to form a ring-like structure. Okay, so this is the Ku protein heterodimer. This is both of them put together now. They've formed this ring. And the idea is that this ring is going to sort of thread onto the broken end of the DNA, basically. So you're going to put one of these rings around this piece of DNA here. Okay, and then one of these rings is going to thread onto this free end of the DNA here as well. Okay, so here are the Ku protein heterodimers which are going to associate with the free ends of the DNA at this double-strand break. Now, they then are going to bind to another protein which is then going to come and bind on top of them. Okay, so each one of these Ku protein heterodimers is going to get another protein associating on top of it. And this is actually a kinase enzyme, so I regret not actually drawing it with an active site the way I usually draw kinase enzymes. Okay, so there's our active site here. Okay, right, so I'll colour these ones in vivid purple here. Okay, so here's one of them, and here is the other one associated on top of this second strand over here. And this time we do have to draw these complexes for both strands, and you'll see why in a moment. Okay, because obviously when we're going to join them back together we will need both strands in the picture. Okay, right. So, what is this pink blob here called? This is called the DNA, and then you call it the DNA PK, which stands for protein kinase, and then you put a CS for catalytic subunit. Okay, so this is the DNA protein kinase, that's the PK, okay, and then the CS is for catalytic subunit, okay, and this is a serine threonine kinase enzyme once again. Okay, now, So, this is the catalytic subunit of the DNA protein kinase. So what is the full DNA protein kinase? Well, this assembly that we now have here, consisting of the DNA protein kinase catalytic subunit along with the Ku protein heterodimer, this entire complex of these three proteins. So we have the DNA protein kinase, I've missed three letters out. The DNA protein kinase catalytic subunit, along with the Ku protein 70 and the Ku protein 80, the entire assembly there is now known as the DNA protein kinase holoenzyme. Okay, so when we talk about holoenzymes, what that means is that it's an enzyme made up of multiple proteins. Okay, right. Now, the binding of the DNA protein kinase catalytic subunit to the Ku70, Ku80 heterodimer changes its activity. Okay, so it is true to say that it's only really an active functional protein kinase enzyme once it is associated with the Ku70 and the Ku80. So that's the logic in calling this the DNA protein kinase holoenzyme. Okay, right. Now, what then is going to come and associate on top of the DNA protein kinase catalytic subunit? Well, another enzyme is going to come and associate on top up there. Okay, and this next protein is something known as Artemis. Okay, so this is Artemis. Okay, and this is going to be a nuclease enzyme. It's going to break down DNA. Okay, so let's colour this in red here. Okay, so now on top of the whole DNA protein kinase holoenzyme, we are getting Artemis associating. Okay, what's then going to happen is the catalytic subunit of the DNA protein kinase enzyme, which is only active now that it's bound to the Ku heterodimer here, is now going to phosphorylate residues within the Artemis protein. Okay, and that is going to activate the Artemis enzyme. Okay, and now what's going to happen is the Artemis enzyme is basically going to clear this double strand break up. Okay, so it's going to make it a nice clean break. So what it's going to do is it's going to get rid of these overhangs. It's going to get rid of the single-stranded portions of DNA. So it's going to say, get rid of this and get rid of this. And what you'll end up with then is a nice clean break here now. Okay? The final thing that now is going to come and happen is a complex of 3 separate proteins is going to come in and bind here. Okay, and I'll show this complex here. And this is a complex of 3 separate proteins. One of the most important proteins here, which I'll have as this big central one here, which I'll colour in, I think, in orange. This is going to be the ligase 4 enzyme. Okay, I might even try and write that in there. So this is ligase 4, and because it's terribly important, we don't just write 4, we write 4 in Roman numerals. Okay, so there's ligase 4. This enzyme up here, or rather this protein up here, is something known as the XRCC4. Okay, which isn't some terrible Roman numeral, but in fact stands for the X-ray repair cross-complementary protein 4. Okay, so the X is for X-ray, the R is for repair, the CC is then for cross-complementary. Okay, and then it's the X-ray repair cross-complementary protein 4. Okay, so that's XRCC4. Okay, so one of those proteins up there is XRCC4. So we'll have XRCC4 in green here. Okay, and this can be this one that's on top of ligase 4 up here. Okay, so this is the XRCC4 here. Okay, and then that final protein here is something known as XLF, which stands for XRCC4 like factor. So I'll put this down here. So XLF, which stands for XRCC4. Okay, and then it's similar to XRCC4, so we call it the XRCC4 like factor, or XLF for short. Okay, so we've coloured in XRCC4 in green, so we'll underline its name in green, and we'll colour in the XLF, the XRCC4-like factor, in blue, so we'll underline it in blue, and now we'll colour it in. Now, it's notable that the XLF also has another name, okay? Its other name is the cernunnos, okay, protein. So it's also called cernunnos protein. So that's another name for this XLF protein. Right, now this complex of ligase 4 along with XRCC4 and XLF, this is now going to religate the two blunt ends that we've now got here together. Okay, so it's going to bring this blunt end and this blunt end together. So they're nice clean blunt ends now. It's going to religate them together. Okay. But have we got back our original DNA? Absolutely not. We have lost what was ever in the middle here. Okay, the portions that corresponded to these overhangs has completely been lost. Okay, this could potentially cause frame shifts and therefore could completely change the way that the ribosome reads the downstream genetic code. Obviously, once this has actually been turned into a piece of mRNA. So it could cause a devastating mutation, basically. So non-homologous end joining is not a brilliant solution to the problem of DNA damage. There is a better way of doing this, which is homologous recombination. However, this is the more common way that the cell actually repairs damage in the DNA, such as double-strand breaks. So the overall message is try and avoid ionizing radiation, okay, because even though your cell can repair, DNA being chopped in half, the DNA is never the same again after it when it does it through non-homologous end recombination. End joining. Okay, so potentially it can lead to mutations in the genes, and this may work towards, it may be one of the factors that works towards tumorigenesis. Okay, so that now concludes our discussion of non-homologous end joining, NHEJ.